Hello, biology students. Mrs. Lewis here at West County High School. We're on Chapter 7 and beginning Section 2, all about the plasma membrane. So the main idea of this section is the plasma membrane helps to maintain a cell's homeostasis. So here you have a picture of a plasma membrane, and in your textbook you also have the same picture. So what you need to do is write down function of the plasma membrane and the page number you can find on it. And underneath this you need to see go to book and look at the picture of a plasma membrane, because this will be on your test. The process of maintaining balance in an organism's internal environment is called homeostasis, and one of the structures responsible for homeostasis is the plasma membrane. It forms a thin, flexible boundary between a cell and its environment. So what's the function of it? Well, plasma membranes have something called selective permeability, meaning they allow some substances to pass through while keeping others out. And they can keep control of how, when, and how much of various substances enter and leave a cell, and that depends on the structure of the plasma membrane. Inside this picture, you can see that water comes in and it leaves, oxygen comes in, carbon dioxide goes out, glucose goes in, and wastes go out. So the structure of the plasma membrane, write this down, and it's correlating a textbook page number. The phospholipid bilayer. The plasma membrane is composed of that, and there's actually two layers of phospholipids, and they're arranged to allow the membrane to exist in a watery environment. The phospholipid bilayer has polar heads facing outside and nonpolar tails facing inside the cell, allowing the membrane to keep the internal and external environment separate. So if we take a look at the picture, here are the heads, these little round balls. These are the polar heads, and they're at the top as well as the bottom. And so if we think of the polar heads as two slices of bread, okay, and we're going to make a sandwich. So then the peanut butter on the inside would be these tails. Now at this point, it's important to note that you could either draw this and label it, and if you draw it, I would highly suggest you color it, or you can just write down the textbook page number that you can find this on and just say, this will be on my test. So structure of the plasma membrane, other components are the plasma membrane. It also controls cholesterol, proteins, and carbohydrates. Some proteins, called receptors, transmit signals to the inside of the cell, while some proteins serve as support structures for the membrane itself. There's something particular called a transport protein, and it moves needed substances and waste through the membrane. So if I were to slice this open of plasma membrane and really dive in with my microscope and take a look, I will see here I have a tunnel, or say a doorway, in and out. And this structure allows for food and waste to go back and forth. Now it's important to note that cholesterol prevents the fatty acid tails, these right here. Okay, it's important to know that cholesterol prevents those from sticking together. Okay, so cholesterol contributes to the fluidity of the membrane, meaning it keeps it nice and moving. Kind of think of a sheet waving in, or a flag waving in the wind. You want that to stay pliable and malleable. You don't want it to become kind of rigid and solid. Now you'll notice this structure right here. This is a carbohydrate, okay, and it defines the cell's characteristics, and it also helps cells identify chemical signals. The fluid mosaic model, so important and most definitely on your test, describes the phospholipids in the bilayer as a sea in which other components can float and move around. The different substances in the plasma membrane create a pattern or mosaic on the surface of the cell.
Have Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy, though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our favorite examples of active transport is the sodium-potassium pump. So that's definitely something worth checking out. There's other times a cell needs to exert energy for transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide. If you check out our biomolecule video, that's a large carbohydrate. Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside, and this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is kind of a general term. There are different types of endocytosis depending on how that cell is bringing substances inside. Amoebas, for example, rely on a form of endocytosis. In this example, pseudopods stretch out around what they are going to engulf, and then it pulls it into a vacuole. And there are other forms too, such as the fancy receptor-mediated endocytosis. This is where cells can be very, very, very picky on what's coming in because the incoming substances actually have to bind to receptors to even get in. Or pinocytosis, this allows cells to take in fluids. So to the Google to find out more details of the different types of endocytosis. Exocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall, but plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So just to let you know, this is a video that you're going to be viewing a lot because it's going to cover this entire unit, several chapters worth of information. It's not going to go away. So anything they mention in the video that will be found in chapter 7 will also be found in chapter 8, will also be found in chapter 9. And when we're past cells and we're talking about genetic material, we will still be coming back and talking about it. So I can't encourage you enough to pay attention to the videos because you never know when you're going to get a quiz.